When we hear the term mangroves, we think wet, smelly, muddy shorelines littered with trees, right? However, mangrove ecosystems are extremely complex environments that involve the interactions of multiple systematic groups, including bacteria, fungi, microalgae, invertebrates, birds, and mammals. And amazingly, up to 75% of the world's tropical and subtropical coastlines are dominated by mangroves. Due to the low levels of wave energy, shallow depth, weak currents, and low winds, mangroves provide a sheltered nursery for juvenile and small fish and invertebrate species to shelter and grow. Furthermore, any material that falls from the mangrove trees begins the degrading process, and colonies of fungi and bacteria consume the fallen material. The degrading process turns the vegetative material into nutrient-rich, high-energy matter called detritus. Detritus is a source of nutrients for many organisms living in the mangrove ecosystem, including crustaceans, mollusks, and small fish. Detritus is also transported out of the mangroves through tides and rainwater runoff, and becomes a source of nutrients for organisms living in surrounding coastal waters. There is significant literature to suggest that crabs are actively feeding on the fallen vegetative material. Twilly et al. In 1997 discovered that the mangrove crab, Eucides occidentalis, can harvest the equivalent of 1 day's fallen leaves in only 1 hour. It is well known that mangrove crabs are having a direct impact on the amount of detritus being retained within the mangrove systems. However, it is still uncertain about how much impact the crabs are actually having. To date, methods for estimating crab density have been unsatisfactory. Methods such as pitfalls, burrow counts, and visual census have not been able to accurately count for crab abundance. And for this reason, in this study, a group of undergraduate students endeavoured to accurately quantify crab abundance whilst testing for a preference in food choice using a completely non-invasive counting method. The study took place at Tower Point Nature Reserve in Botany Bay, Australia. The location was dominated by two main species of mangrove: Avicennia marina, the grey mangrove, which is characterized by its upright roots called pneumatophores, and the river mangrove, Ageroceras carniculatum. Tower Point is a unique location as it's located right next to Sydney's CBD and is fringing right on Sydney's main airport, yet remains to be host to 50% of Sydney's remaining mangroves and is the last remaining salt marsh in Sydney. Tower Point is a complete wetland system containing mangroves, salt marshes, extensive seagrass beds, sandflats, mudflats, and small patches of rainforest. The various ecosystems at Tower play host to a diverse range of plants, crustaceans, mollusks, mammals, and numerous rare and endangered bird species. Some have migrated 12,000 kilometers from Siberia, such as the little tern. We decided that the use of GoPro cameras was going to be the most non-invasive method of counting crabs, while still allowing us to have analytical power and efficiently quantifying if the crabs have a preferential food type. A 1 meter quad was divided into 64 small squares using high visibility brickies line. Within the 1 meter square were 4 smaller quads that we randomly allocated 4 food types to. These food types were mangrove roots, seagrass rack, mangrove leaves, and a blank control. By dividing the quad into 4, we were able to maximize replication. A PVC frame was made for the GoPros and the cameras were set up to take photos every 30 seconds for 2 hours. We also counted hole density prior to the filming. We set up a camera trap near the tidal creek close to the shoreline and another camera trap along the levee that divides the salt marsh and the mangroves. We believe there could be a site-specific difference between the root density, shelter and composition of the surrounding plants. As we had access to a third GoPro, we also set up feeding tubs. By firstly catching a number of crabs, we then lined 6 tubs with 2 to 4 centimeters of sand and placed the 4 food types across 8 cells within the tubs. 6 to 8 crabs were placed in each tub and the cameras were left for 2 hours taking a photo every 30 seconds. Each of the 4 team members were initially given 10 photos from the levee site and an additional 8 photos from the creek site. Photos were split 10 minutes apart to avoid double counting the same crab. Food type, crab abundance, photo number, hole abundance, and the idea was recorded. for each of the 64 cells within the 1 meter square quad. Across the two sites, we identified a total of 4 species of mangrove crabs. The most common was the Helioesis cordiformis. The Levy site produced some compelling data. We can see there were significantly more crabs in the cells with no food source. This may be due to the fact that food sources were blocking the crab holes that would have otherwise contained crabs we would have counted. Seagrass wrack was the least favoured of the 4 food types. There was a very low correlation between hole density and crab abundance, which would suggest that our results are significant. There was no significant difference between any of the food sources at the creek site. If we had analysed more images, this result may have changed. There was no significant results from the feeding tubs, however the crabs were observed trying to escape or hide from the sun during the filming process, so future studies should look to avoid containment methods when working with crabs. Although this was only a pilot study, I believe we were highly successful, although we did not find significant results. Firstly, we were successful in developing a new means to observe crabs without disturbance. This discrete method can be applied to many other species, whether it be birds, mammals, or other invertebrates, and this method also has higher analytical power. Further studies would incorporate greater emphasis on species-specific traits within and across sites, as well as noting life stages and gender differences. High replication of image analysis and camera traps would yield greater results, and I believe if we repeat the study each year, we'll be able to understand responses to sea level rise. And sea level rise combined with constant anthropogenic impacts are the 2 biggest selective pressures on the myriad of species that reside at Tower Point today. From dredging the channel for shipping lanes, to land clearing and bay developments for the 3rd runway, all of these operations have a major effect on the wave energy, tide cycles and erosion rates of nearby locations, such as Tower Point. I believe the study has shown that we can utilize modern technology such as GoPros and drones to acquire stronger statistical power, as well as introducing a visual tool that may be more effective in raising awareness compared to a journal.